So moving on to our next panel, this one is actually really also very, very interesting because few fields in modern medicine have generated more public excitement than the quest to extend human life. As we get older and the cells in our bodies decrease in vitality, the mind often slows and the body starts to break down. I tell people I still have a photographic memory, I just don't have same-day service anymore. You have to be of a certain age to even remember what the heck that means now with digital cameras. But what if we could reverse that process, right? What if we could replace our older, poorly functioning and dying cells with fresh, vital ones? Or what if we could alter our DNA to actually slow the aging process in the first place? Some researchers believe that's actually doable. Please turn your attention to this video.
I feel tremendous. I'm ready to take all the world.
In Ron Howard's movie Cocoon, senior citizens are rejuvenated by bathing in a pool storing alien beings in pods. Eventually, the aliens offer the elders much more than newfound vitality.
We'll never be sick, we won't get any older, and we won't ever die.
It's a metaphorical take on the fountain of youth, something humans have sought for millennia to no avail. Yet there's a growing handful of people who seem to have found a way to live not just far beyond their predicted lifespan, but to also remain vital into their 10th decade of life. Dr. Barzilai is studying a group of these exceptionally healthy centenarians, which became part of another Ron Howard film called The Age of Aging for the National Geographic Channel.
So nice meeting you. How are you?
He believes they're a model for how we can all age.
Come on in, fellas.
Hello.
Hi, how are you?
How are you?
One of the interesting things with those centenarians is to see how they interact with the environment. And we thought, hey, maybe they do all the right things. Do you eat something special? Do you...
No, I try to keep a healthy diet.
What about exercise?
Exercise, I walked about a quarter of a mile for breakfast.
I swam every day.
So my body has always been activated.
And when you look at the population, you find almost the opposite.
I go just about every afternoon to Dunkin' Donut, and I have coffee there and a Boston cream, that sort of thing.
Your eating habits are not necessarily healthy.
That's true.
I smoked for
Minus 14,
50,
right?
so 50 years.
50% of them are obese, 50% of them do not exercise, 60% of the men and 30% of the women are smoking. So it's in spite of all that, that they have some protection that allows them easily to get to age 100. What we do find that they have is genes that are protecting them against anything that's thrown their way.
Dr. Barzilai believes his centenarians have genetic mutations that seem to slow their biological aging. He also believes there's a medication that may be able to delay the effects of aging in the rest of us. Metformin. Really? A well-known, well-studied, inexpensive, FDA-approved diabetes drug that will be used as a tool to change FDA indications specifically to target aging. It's been said that having kids later in life keeps you young, and research suggests that being around young people can keep you feeling and acting young. Well, it turns out there may be a cellular version of all that. It comes from experiments done in the Rutgers, New Jersey Medical School lab of Dr. Pranela Rameshwar. She's found that taking stem cells from young people and incubating them with stem cells from older people has a remarkable rejuvenating effect on the older cells.
We separate these cells by a membrane. And what we noticed is that the old cells were communicating with the new cells through that membrane and telling them, perhaps, restore me. And the young cells, in turn, were releasing factors. And that was getting through the membrane, entering the old cells. And those old cells, suddenly, when we do the test, they were functionally similar as the young cells with time.
Why is that important for aging? If those stem cells come from bone marrow, the revitalized cells would generate a more competent immune system. That would enable an older person to ward off the often lethal viral and bacterial infections of the elderly. Just as importantly is the impact a reinvigorated immune system could have on cancer.
Older people tend to have cancer at a higher rate, but now you have a competent immune system with a better surveillance system to get rid of those cancer cells before they become a clinical tumor.
While these findings argue for banking your stem cells while you're young so that they can be used to rejuvenate your cells later in life, experiments are currently underway to identify the factors that stem cells use for communication and rejuvenation.
So we think we know what factor is produced by the young cells to get into the old cells. We may be able to isolate that factor and make it into a drug. Perhaps it could be just made into a factor as a drug, injected, or maybe we may need the aid of other stem cells to deliver so it goes exactly where we wanted them to go.
However this rejuvenating factor is delivered, the goal is not just to contribute to a longer life, but also a healthier life. A related but somewhat different approach to using stem cells for rejuvenation is being taken at Human Longevity, Inc.
Right now, we're carefully studying the quality and the quantity of stem cells in our different tissues and our organs as a function of age, and we are treating those changes by redelivering young stem cells. And what we're finding is that those young stem cells have the ability to improve the quality and the functionality of the whole stem cell population in the organ or the tissue.
The problem is, where do you get these younger vital stem cells to give to an aging population? Cells from young donors present rejection issues, and in any case, there likely wouldn't be enough donors to satisfy the demand. Turns out that healthy human placentas are an abundant source of stem cells, or technically very early progenitor cells.
What our clinical research has shown us is that placental stem cells are one size fits all, and that I can give a stem cell from one placenta into someone who's not even related to that donor, and they take up and do their job perfectly well. A placental cell is the perfect donor material, which means there's hope for you and me. We didn't have our stem cells stored away, but we have the benefit of using stem cells that are normally going to be disposed of.
But however this rejuvenating effect is achieved, drugs, cytokines, donor stem cells, or other ways still being developed, the goal must be improving the quality of years in a life, not just the quantity. Otherwise, longer lifespans would be disastrous for society.
We're using up so much of the planet's resources now. We have over 1 billion people in poverty. So just adding more people to the planet by having them live longer is not a good society goal. Having everybody have an increased healthy lifespan increases productivity, and hopefully we can move more into a sustainable kind of living.
I couldn't play violin when I was young. Apparently our videos could use a little I think our videos can use some younger stem cells in them somewhere along the line there. To discuss this really fascinating field, let me start out by introducing our very distinguished panel here. To my far left is Dr. Ronald DePinho, is the president of the University of Texas MD Anderson Cancer Center, and I might point out my pronunciation is better than the vice president's. Immediately to my left, you'll recognize him as Dr. Robert Hariri, the co-founder and president of Human Longevity, Inc. To my right is Dr. Pranela Rameshwar, professor in the Department of Medicine and a division of Hematology, Oncology at the Rutgers, New Jersey Medical School. And then to the far right, Dr. Barzilai is the director of the Institute for Aging Research at the Albert Einstein College of Medicine and director of the Paul F. Glenn Center and National Institute of Health's Nathan Schock Centers of Excellence. Now, I'm going to ask each of you to give us a brief presentation, and I just realized that as I started doing this, I'm not sure what order our slides are loaded in. Did we find out?
From that end this way.
Straight across. Near, you're up first.
Terrific. First of all, thank you so much. Thank you for the Vatican. Thank you for
Principessa Robin for her what she's doing, and it's been a great time here. Look at the title that I chose. Look, we don't like aging because for us, aging means chronic debilitating age-related diseases. We don't like aging because after the age of 60, there's a takeoff of how many people have more than 3 diseases and then 3 treatments for those diseases. But as a community in the last decade, we have been able to extend health span, and also lifespan. A variety of models. We have been doing it with worms, with flies, with mice, with rats, with primates. And we've been doing genetic tools and environmental tools and drugs. And some of those drugs are in human use. So for us, the moonshot equivalent is we think that we can now spend 80 years in order to get to the biological age of 60. Those are what our preclinical trial have shown in animals. And those were preclinical trials that were in multi-centers. And so we are very excited about that. I want to say something about, because the people who are most concerned with our optimism are some of you guys. I can see your faces, the oncologists. And I just realized why. Because your patient have his own genome that is unique, and it has the genome, the genetics and epigenetics of the tumors, and then the tumors are clonals, and none of the tumor looks like anyone else's tumor in the world. This is huge. It's very different with aging because aging is conserved, evolutionary. If you remember those 3 bear slides and all the species, every species you give the treatment and they behave very similarly. So we come more optimistic. Now, I'll go in very few slides. I have 5 slides that I want to show you what are we really trying to say here. What's the impact that we're trying to do and how we're trying to do that. So this slide simply shows the relationship between age and aging for a variety of age-related diseases. And you have your favorite, but the tops are heart disease and cancer and diabetes and Alzheimer's. But notice that it's a log scale. When you go from 30 to 100 to 80, your chances of getting those diseases go from 1 to 1,000. Okay, when we're talking about cholesterol and heart disease, it's a 3-fold risk. The National Heart and Lung don't put aging as a risk, okay? But it's the same risk for all the other diseases. Look how the lines are parallel. And so when we looked at this graph, we said something that's obvious for us, that all we can hope is, unless we target aging itself, all we can hope is that we switch one disease for another. I mean, if you go to the emergency room and have a heart attack and get a stent, it's a local treatment, okay? We are not going to change your rate of aging. What happens to those people? Within 2 years, they're getting cancer or Alzheimer's or heart disease because we never treated this disease. Imagine you'll cure cancer. What disease are we going to get? So this is the rationale that the only way we can make an impact, a big impact, is actually to target aging. So the question is, can we extend health span? And you just saw our centenarians, and the question is really, have they been sick since they're 60, 70, 80 years old, and they just crawled into this old age? Or have they been healthier? I mean, are they a good model? And this is the answer. When we look at control and centenarians, and what we're measuring here is outcomes of age-related disease, you see that it's not only that they lived longer, but they got their diseases 20, 30 years after the control. So in this case, lifespan and health span are associated, but in no way I have any desire to extend lifespan.
And when we'll have this medication in the commercial, we'll apologize for the longevity part. But it's really the health span that we're trying to target. There's another important thing that we learned from those centenarians, and that is not only did they get the disease later, but when they got the disease, there was a contraction of morbidity. They didn't spend the last 5-7 years of their life being sick. Rather, they spend 5 months or less of their end of their life being sick. And this actually has been measured by the CDC. It's been doing it every year. This is a slide from 1993. And what it shows you is the medical expenses in the last 2 years of life. Okay? And you see that if you die after the age of 100, your medical costs are a third of if you die between 60 and 70. There's a huge longevity dividend here. It's estimated that if we can imitate that, even if we'll extend health span a little bit, it's a $7 trillion saving until the 2050, and there's a lot of literature about it. So what are we going to do? Well, there is this drug that was mentioned, metformin. It's the first line of drug for type 2 diabetes. It's been around for 60 years. Its safety record, there's nothing we don't know about it. It's generic, okay? It's cheap. But we know other things about metformin. First of all, when you give it to nematodes or to mice, they live healthier and they live longer. So it's in the midst of the biology of aging. By the way, I'm not touching about the biology of aging. Ron DePinho will give an example of that. Maybe it's Ron DePinto because of the telomeres. Now I'm thinking, maybe he didn't do a mistake. You can call me Biden. Not only that, when you do control studies, metformin prevents diabetes in people without diabetes. Metformin prevents cardiovascular disease in many studies in diabetic patients. In association studies, metformin, people on metformin have 30% less cancers, all cancers except prostate cancer. The NCI is 100 grams on trying to look what metformin does in different stages for prevention and for metastasis and for other things. Metformin prevents cognitive decline even in non-diabetic individual. But really, I think the coolest data is from this. It's kind of a phase 4 trial because metformin is on everywhere. So you can go to the pharmacy and identify people on metformin and see what happens, in this case, to their mortality. And you see here 4 groups, so I'll tell you it's very easy to understand. The blue group in the lower, that the mortality is increased most, are diabetic patients who are treated with sulfonylurea drug. That's the other drug of choice for diabetes. And they have mortality much more than their control group, which is in red. By the way, there are 200,000 people in this study, okay, that were followed. The green group is the metformin group. So the people on metformin, first of all, have diabetes. Second, they are more obese than their control group. Third, they have more diseases to start with than the control group. But their mortality is 17% less than the control group, in spite of everything. It really shows you that metformin has a very strong effect on aging, even on mortality. So you're asking, why are we doing the studies? I just told you all the studies have been done. And I'll tell you honestly, I have no interest in doing this study. There's something cool that I'm interested in the study in the sense, well, I'll tell you it in a second, but this is the reason I'm doing this study.
You're going to do that quickly though, right?
Yeah, I'm almost done.
Okay.
The FDA has been our friends. The FDA understands exactly what we did, what we're trying to do. We're a bunch of scientists. We're not representing any pharmaceutical. We never discussed the drug metformin because it's in the FDA, in the FDA page. With the FDA, we've discussed what study can we do in order to get an approval that will reflect aging or the comorbidities of aging. We don't want to use necessarily the word aging, but the comorbidities of aging. And this is really the main reason we're trying to do it, because there'll be next generation drugs. If we don't do it, pharmaceuticals will not develop the drug, because the healthcare provider wouldn't pay people to get the drug. We have to have the FDA in order for the pharmaceuticals to develop better next generation drugs like they've done elsewhere. We also will provide the template for those people to know what we're doing. It's not much. It takes much less than any drug that you'll develop for diabetes, cancer, or other things. And of course, we also want to make sure that the people who are in this anti-aging therapy know what studies they have to do in order for people to believe them, to believe in what they're doing. So I'm sorry if I'm late. I'll stop here. Thank you very much.
Thank you. Thank you, Nir. So we have... Our next two presenters are going to take a somewhat different approach, a cellular approach here to anti-aging, and we'll begin with Dr. Rameshwar.
Okay, so the last 2 days of this meeting, you heard about boosting the immune system, and that is help to get rid of cancer. So the approach that we take in this aging study is to go to the source of this immune system, and that source, if you look on your left, is the bone marrow. And I use this diagram to show you what we're dealing with. We're dealing with a very complex organ. And this organ has been studied for a decade. And it's still not known exactly how this organ is organized. Just to give you an example, in the far left, just a few years ago, we thought the hematopoietic stem cell, which are the cells that give rise to all your blood system, was located close to the bone area. Now we know that that's not true. The bone marrow is highly vascularized, and normally when you hear something is vascularized, you expect to be oxygenated, but that's not true. The bone marrow, despite the oxygenation, the vascularization, the oxygen level is very low, and that's good for stem cells. Stem cells like low oxygen, they survive very well in oxygen. So a lot of the new data is saying that the hematopoiesis occur next to the vascular area. So what's the consequence of having a functional bone marrow? The 10 to the 12 cells are emitted from the bone marrow into the periphery. And if you look on the right, those cells end up in the lymphatic. What is the lymphatic? Is the connection of the immune system. That's the system that can depict when there's an infection or there's a cancer. So what happened in aging is that if you go back on your left, is that entire bone marrow microenvironment become dysfunctional, and the oxygen level is different. So our aim is to get that back, take a 70-year-old and try as much as we can to get it functioning as a 35-year-old. It's not...
It will. It takes
a
second.
It takes a while. Now, this is a simplistic view of how the stem cell differentiate. Now, if you look at the top, is the hematopoietic stem cell, and that gives rise to other stem cell, and then you start differentiating into those cells that you know as your blood cells or immune cells. But I want you to pay attention to the far CLP. That's the lymphoid system, and on your left of that system is the myeloid system. The problem, as you age, you have more myeloid cells than lymphoid cells. And this is serious, and the reason it's serious is because if you look below CLP, that's the cell that make your B cells, your T cells, and your natural killer cells. And if you remember during the last 2 days, you hear how important those cells are. You need your natural killer cells. That's the innate immune system that surveil to get rid of any tumor that may be developing. You need B cells if you have a vaccine. Everyone says older people should have a vaccine, but we don't have lymphoid cells. So by restoring, now I'm going to use this word restoration a lot. So what I mean by restoration is taking an old hematopoietic system and try to make it into a young hematopoietic functional hematopoietic system. So in older people, because you don't have those lymphoid system, you end up having reduced immune surveillance. You can respond to infection. You can respond very well to a vaccine. But more importantly, everyone knows someone who is older have a myeloid leukemia or some myeloid dysfunction. So if we restore this, we'll be able to prevent perhaps leukemia. At the same time, we have good NK cells. So because let me show you some of the data. So what we've done, we have started with working in the lab. Now, in your left, you see there's a co-culture system. Now, let me tell you what MPB. So someone yesterday or the day before described how you mobilize peripheral blood to do bone marrow transplant. This is an expensive procedure because you inject someone with a cytokine called GCSF, and you pretty much have an in vivo expansion of your hematopoietic stem cell, get into the periphery, and you're in a pheresis machine and you harvest those cells. Now, GCSF is off patent, but it's still very expensive. We have also tried umbilical cord blood, and I'll get to that in a second. And at the bottom, we have the age itself. And so there's a communication between those two cells. And the reason I have a double yellow arrow is because there's more communication, the young cells telling the aged cells something, said, okay, go and change. And the aged cells is also talking to the young cell. So what did we observe? And these are the highlights of what we observed. We observed significant functional natural killer cell. And the reason I stress functional, it would be useless if it's only by a biomarker. Without a function, it means nothing. They're functioning. I can't talk about what these restorative factors are for obvious reason, but we think we know what the restorative factors are. When we started this project, we were doing this co-culture for 28 days, and as you know, that will not be good for the FDA. We've been able to reduce that time to 7 days and have even better restoration, and that's really amazing. The FDA like anything less than 2 weeks, so they'll be happy with this. Okay, so the reason I choose this slide is because the most important thing, if we're going to change the hematopoietic system to make good immune system, we have to pay attention to the hematopoietic stem cell. So look at the graph on your right, and pay attention to the green and the red lines. And look between day 6 and day 10. As you can see, the red, no restoration. The green is where you have the restoration. It is significant improvement. Now, one of the things we need to do, and all of you may be asking, why don't you use umbilical cord blood? If you live in New Jersey, the state pays to store umbilical cord blood, and we get it for free. So as soon as the bank has the blood, they call us and we drive and pick the blood up. So we've been able to test 12 different umbilical cord blood. What we found is that consistently the mobilized peripheral blood can restore. But we did not find that with the umbilical cord blood. So we decided why. We look at the different things, the time of delivery, the volume, because there's an ethical regulation of how much blood you can take from the cord, because the cord belongs to the baby, not to the mom. But we didn't find any difference. So we can never know the health of the mother. They will not allow that. So that could be an issue. So we decided to look to see what may be the reason. Now the CD34 is a biomarker for all hematopoietic stem cell in human and progenitor. That really matter. So the amount of CD34 we can harvest in the cord blood will predict if that cord blood could restore. And that's consistent with the mobilized peripheral blood, showing that those CD34 positive cells are very important in the restoration process. Now, one of the things that you always have to worry about is when you're restoring with someone else, because all of you have different match. If someone is doing a bone marrow transplant, none of you probably match with each other. So that's called the MHC class molecule. So it could be we'll restore and we put it back into that patient and the patient start rejecting it because now they have the MHC of a donor. If you look at the graph on the right, for the scientists in the audience, that's a mixed lymphocyte reaction. For the non-scientists, all is showing that it's a safe. to go through and do the restoration with someone who is unmatched from a different type. Now, this is my last slide, or, well, second to last. Now, this is in vitro. We need to go to in vivo. So we choose the most immunodeficient mice, called NSG mice. We humanize them with old mobilized peripheral blood. Now we're seeing if we can humanize them with the non-restored cells, then come in with the restored cells and see if we can mobilize them. If you look at the bottom left, we are able to now humanize those mice with lymphoid cell, because that's the important thing. So this is so now ongoing, as we speak right now, we have restored cell in these NSG mice. And I just want to tell you, we've been very successful with this in vivo restoration. We can humanize 100% of these mice, which I did not expect. Because normally you get about 75%. So we're very happy that in a couple weeks, we'll be able to say we can do in vivo restoration. So I just want to conclude with some key points. The key points that we can mobilize with young, mobilized peripheral blood. We can restore, with cord blood. There is consistency more with mobilized peripheral blood, and we have to find out why we can't mobilize with all the umbilical cord blood. And we really want to do the umbilical cord blood because that would be cheaper and have the technology available to everyone, regardless if you have money or not. The mitochondrial function of the cord blood seems to be important, the CD34 positive cells. We do not need a match. We can take anyone. The procedure is safe. We have no evidence, and we've really looked, because we're also part of the lab is a cancer lab. So we're very mindful of looking for transformation. There's no evidence of transformation. The humanization of the mice is successful. In a couple of weeks, we will know if we can actually restore and how long the restoration process would occur. We think we're within the FDA guidelines. Now, one thing I want to say that this work is funded by the Barzilai Life Sciences. So this is free from him. He gives me full academic freedom to do this work. He never interferes. And normally in the last slide, I would use this slide to say who was doing the work. But you hear a lot about combining public-private partnership, academic biotech. I think I work a lot with company, and this is one of the few companies I think it worked really well. Because Dr. Barzilai funded this project, he give me full academic freedom. The students learn, and what we do in the lab will also benefit him in the long term as he takes this project to the clinic. So the last thing I want to say is that you might be asking, where are we going? Do we really have anything here? So this is now the academician side of me, not Dr. Barzilai or his team saying this. I think he's not so far for cellular pharmaceutical. And then as we understand the mechanism, it will be biopharmaceutical. And then hopefully he'll be able to use stem cell to deliver the biopharmaceutical. And I want to thank you.
All right, good. Thank you, Pranela. So I know we have lots of questions here that we want to get to, so Bob and Ron have promised me to go quickly.
Yes, and
Dr. Robert Hariri, I'm sorry.
I have the most slides in all of them, but I'm going to go really, really fast because you are all aging before my very eyes. So I've spent about 20 years in this business, building first a fairly diversified regenerative medicine company that has a number of products in clinical programs, everything from treating autoimmune diseases to treating things like age-related blood vessel damage. And the fields that you've heard about since you've been here, like immuno-oncology, are central to the things we've been doing. But for the last year and a half, I've been really interested in focusing on the fact that age, as one of the greatest and most important risk factors for all diseases, is something that we potentially have an opportunity to impact using cellular medicine. And what I'm going to try and convince you all is that stem cells, aside from all of the miraculous things you have seen them do over the last 2 days, I believe that stem cells may in fact serve as the fountain of youth and allow us to retain high quality, high performing tissues and organs as we age. So stem cells do something that I like to call fueling the regenerative engine of our body. So stem cells are resident in all of our tissues. They drive the natural process of renovation and repopulation and sort of restructuring our organs and tissues in response to disease and injury. And they also, because they reside in a form that has the full transcribable genome accessible to it, preserves what I like to call the synthetic repertoire of the cell. Now, what a cell is, it's a factory. It produces proteins and other substances which help create the structure and function of all your organs and tissues. It turns out that during the process of going from a stem cell to going to a mature, specialized cell, a series of events occur which silence part of that genome. So your biological software, which resides in the nucleus of your cell, encodes for all of those different molecules that make you who you are and function how you do. The fact is that the process of going from a stem cell to, for example, a mature neuron actually turns off some of those genes that aren't necessary for that specialized form. And it turns out that the ratio of undifferentiated cells that have that full synthetic repertoire to differentiated specialized cells has an impact on the quality and functionality of your organs and your tissues as you age. In order to look at this, we actually stumbled upon some interesting observations made from our colleagues in the plastic surgery world. Now, whether by design or by pure luck, plastic surgeons looking to do contour changes, for example in the face, were taking adipocyte and liposuction material and injecting it into the face instead of using synthetic products or collagens or whatnot. And what they found was that these faces were rejuvenated. And they thought, wow, adipocytes, your fat must be really, really a rejuvenating product. In fact, what they were doing is they were transferring some of the stem cells that resided in the small blood vessels that got into the liposuction material. And what actually we saw is if you biopsy the skin of an old person versus the skin of a newborn or a young person, the difference isn't so much in cellular content, it is in the content and diversity of those proteins made by that biological software. So in the skin of an 80-year-old, you can only find about 1/3 of all of the collagens your DNA codes for, whereas in a youthful skin, you see all of them. So we actually believe that the theory behind this is that stem cell differentiation may echo the process of aging, and that the balance of differentiated and undifferentiated cells plays a role in the quality of your tissues and organs and their functionality as you age. In order to look at this, we actually looked at some old data looking at the quantity of stem cells in a tissue like bone marrow as a function of age. And this relatively small data set showed us that stem cell quantities dramatically declined with age. And as you can see in this graph, by the time you're even beyond 50 years of age, the number of cells capable of making blood and blood-forming elements is dramatically less than early in life. Well, we went and looked at this in other tissues. One tissue that I'm particularly interested in, skeletal muscle, shows a profound decline in the number of satellite stem cells. Those are the stem cells responsible for rebuilding your muscle as you live your life, dramatically declines with age, and in fact, that is not related to the actual mass of the muscle. So again, that confirms this ratio problem that I just talked about. And those changes in muscle stem cell populations are echoed in changes in functionality, namely strength and muscular endurance. So this all kind of jives with the theory. And the fact is that the anatomic and physiologic changes that occur with age reflect this continuous renovation of tissues with an ever-contracting reservoir of stem cells that begins to accumulate defects and deficiencies in producing that diverse synthetic repertoire, okay? So the question is, can we restore stem cell quantity and quality? And if so, can we do this clinically, and will it in fact change functionality? And we're in the process of doing that right now, both in preclinical and clinical studies at Human Longevity in San Diego and at my cell therapy company, which is a part of Celgene Corporation. So we actually believe that aging is a stem cell problem. It results from a shift in the balance of undifferentiated to differentiated cells. It disrupts the natural renovation and remodeling process that occurs in tissues, and it corrupts the normal response to injury. I think I won.
Oh, yeah? Watch this, buddy.
Dr. Ronald DePinho.
Yes, sir. I'm going to centrifuge it down and give you the pellet. Okay, here we go. So basically, this is the problem, as Nir mentioned. You basically have a 45% chance of getting Alzheimer's by age 85. 1 in 2 chance cancer if you're a man, 1 in 3 for a woman. It's a problem. And we've got 1.2 billion individuals by 2025 that are going to be over the age of 60, where we have this exponential increase. So I want to convey to you that our knowledge of the circuitry of aging and the biology of aging has now reached a point where we can begin to think about, can we manipulate that process? So I'm going to tell you about the work that we've done on telomeres. These are the caps of chromosomes. They help to maintain your chromosomal integrity. As you age, you accumulate damage at your telomeres, and your telomeres essentially, as a result of cell division and free radicals, become very damaged, and they then are put in a position to activate the aging process. There's an enzyme that's quite low in your cells that normally repairs and maintains the telomeres. So since you don't have sufficient telomerase, you essentially walk this telomere plank as you age, and that then precipitates part of the aging process. So these ends become frayed, and then it activates a circuitry. So we asked the question, if we were to engineer mice where we can artificially toggle on and off telomerase, what would happen? Well, in the off state, these animals aged prematurely. So by the time they were middle-aged, they were the equivalent of, let's say, a person in their 90s. They had diminished cognition, shrunken brains, infertility, thin bones. They were quite feeble, and they had dramatically shortened lifespan. So in that setting, we asked the question, if we were to turn this gene back on and restore telomerase activity in these animals, what would happen? We're expecting a stabilization of the aging process. Instead, we saw a Ponce de Leon-like result. These animals restored their cognition, became fertile, and so on. It was a rather dramatic reversal. It basically teaches us that our tissues retain a remarkable capacity to renew themselves if you remove the underlying instigator of the aging process, which in this case was chronic DNA damage signaling from the telomere. These are stem cells in the mouse brain. That's a normal stem cell reserve. If you have shortened telomeres, what you see is an attrition, a depletion of the stem cells. And now when you flip the switch back on, you actually see a dramatic restoration of the stem cell compartment. Newborn neurons are now regenerated, so on and so forth. And this is true across all organ systems. So essentially, what this teaches us is that this circuitry, which as Mir mentioned, is conserved from worms to humans, our knowledge of it has helped us begin to understand what points of intervention could we deliver to attenuate the signaling flux through this pathway. So if you begin, let's say, at the level of DNA damage, let's say telomere dysfunction, that signals to molecules that are very important in oxidative defense, defense against free radicals, as well as maintaining the powerhouses of your cell, the mitochondria. So as you age, what happens is your ability to fight free radicals diminishes, and your mitochondria become dysfunctional. And so what happens in that context, your free radicals then increase dramatically, and you then have an amplification of DNA damage, so you get into this feed-forward cycle. So this is why we do very well for many decades, and in that final decade of life, we have an accelerated onset of aging because you enter into this death spiral, this feed-forward loop, this signaling loop. So we know a lot about this circuitry, And the telomere reversal, as I mentioned before, teaches us that if you actually begin to break the signaling flux through this circuitry, you can impact on the aging process itself. How's that?
I bet.
All right, I think we're
I'll
done.
get you about 10 seconds.
Thanks. Thank you, Ron. So we don't have a lot of time, so I ask you to keep your answers pretty brief. But Ron, let me start with you since we've got you here first. How do we turn telomerase on in humans, not in mice? What can we do to turn on this repair mechanism?
Well, we're actually working. So one of the things that the reason why it's off is because the transcription, the expression of the gene that's a critical component of the enzyme is low. And so we actually screened 1 million compounds and looked for activators of gene expression, and then further asked in human cells, can it actually restore telomerase activity? And so from 1 million compound screen, we've gotten down to maybe a dozen molecules that we've confirmed and validated, and we're now moving those into more robust chemistry to allow us to increase the potency of those molecules.
And if you want to know what they are, send him a 100 note, 100 euro note, and he'll scribble it on the back of that and keep the 100 euro note. Bob, tell me, we've got stem cells, telomerase, metformin. Are these mutually exclusive approaches to anti-aging, or could they be complementary?
No, actually, I think that this is a complex biological process that requires a somewhat complex solution. And there's no doubt that all of these approaches could be, in fact, used in an organized, synchronized manner. I know for a fact I take metformin because, I believe that there are some clear benefits. Near educated me about the doses you need. I can tell you that before metformin, my eyes were so bad I would not be able to identify the edge sitting in the audience. Now I can. But the fact is that to what Ron's been talking about, there's no doubt that the science around telomerase has been very, very important in aging theory for a while. One of the reasons we're doing stem cell approaches is that's another way to deliver cells that don't have a telomerase, a telomere issue. You're putting in cells that have kind of preserved those telomeric caps. And I think that there are going to be other solutions that may even arise from some of the other factors that Pranela identifies.
So Pranela, tell me then, I know you mentioned that we can get these young cells from cord blood is a good place to get them. And I know you said you're in the process of identifying what these signaling factors are. And by the way, I'm not sure if we got into it very much. It's not just that the young cells are talking to the old cells. The old cells are sending out, like an SOS signal saying, come help me, right?
Right, they are. So one the question, so the cord blood thing is interesting. The mobilized peripheral blood is consistency. The cord blood, not so consistency. I'm trying to find the reason. But there's a possibility that the mobilized peripheral blood may be better because these are adults who have, their cells have been activated. It could be we need some activation. You need more activation. Those cells are active. Cord blood cells are very primitive. So what we're doing, we're continuing to identify these factors with the mobilized peripheral blood, in parallel doing it with the cord blood, and see if it's the same factors are involved, or maybe the mechanism might be different.
But where do you see the anti-aging therapy being cells or identifying these factors, and those factors are then what are purified, manufactured, and given?
It could be both. I think the first thing will be to do cellular pharmaceutical, which will be cells. And then eventually, you continue to identify these factors, make a biopharmaceutical. And now the new thing is like a biopharmaceutical to be delivered with cell therapy. So we could have either the placenta stem cell, maybe the adipose-derived stem cell, bone marrow stem cell. It may have to be the bone marrow stem cell, because we need these, if we're going to deliver these biopharmaceuticals back into the bone marrow. These cells have a memory. I have evidence for this now. We need them to go back to the memory. And and other people who are doing different anti-aging could use adipose-derived cell. They cross the blood-brain barrier. They go to the neuron, as Dr. DePinho was saying, that maybe we can get it telomere by some drug delivery system in those neural stem cells.
Interesting. By the way, I can identify edge too from here. You know, and my eyes aren't that. I haven't been taking the metformin.
I need to put my glasses on.
Okay. Nir, two things that we want to do quickly here. One is I want to make sure that we drive home the idea here that by compressing the amount of time that people, the centenarians, spend actually getting sick is actually a very big saving in healthcare dollars. They don't spend years, they get sick and they die. Tell me, make sure that you drive home why that's a big savings.
So we're talking about third of the medical cost in the last 2 years of life. By the way, when they were 60, 70 years old, they didn't go to the doctor. So it's not that they spend health spend now. But I think that, you know, there is the micro data from my study, and then there's the CDC data, and it's very compelling. You know, you can say maybe the families didn't want to take those guys to the doctors. Good idea. If you're 100 years old, don't go to the doctor. That's why the longest living people in the world live shorter. But I think those are kind of the data. The contraction of morbidity is a major data for us to understand, because this is the translation for the economy.
And finally, and I hesitate because I suspect that the answer might be too long, do we know the mechanism of metformin's longevity effect?
Metformin looks like a super drug, right? It acts on many nodes, including some of what Ron have shown. And for us, the question, no, we don't know the mechanism. The question is it really a super drug, or does it fix something that we don't understand? What is primary in aging? Does it fix it, and then everything comes to be in point? But we really don't know. We know the glucose lowering effect. I'm not sure that that's the longevity effect.
Interesting, very interesting. Lots of questions here, but it looks like we may actually be able to live for quite a while longer. You know, if some of us that are on the back nine, we may actually get a few extra rounds in, right? Thank you, ladies and gentlemen. We're going to take a quick, that's a golf reference for you guys. Thank you. We're going to take a very short break again. Please come on back. Come on back by 4:40, 4:45. Come on back.